So I will get started. So in this presentation, I'm going to introduce a method to predict the disorder flexible linkar regions in protein sequences. The name of this method is DFLpred. DFL stands for disorder flexible linker. So I will start with an introduction of what disordered flexible linkers are. DFLs are regions that serve linkers or spacers between protein domains and between structured intradomain constituents. So this means these regions can stay in between domains and could also be inside domains. DFL is the most annotated non-binding function of disordered proteins. We use the annotations from the DFLpred, which is the largest source of disordered proteins, annotations of disordered proteins. The function of DFL was defined by Professor Dunker in this paper. In 2002. DFLs differ from classical linkers in three ways. First, they have undefined structure because they are disordered. Secondly, they are relatively longer than classical linkers. And as I mentioned before, they could stay inside domains and could stay in between domains. I will show examples for DFLs which are inside domains in the presentation later. We focus on DFLs for two reasons. First, they are functionally important. They help the formation of amyloid fibrils. They link multiple disordered protein binding regions, and they help the movement of structured protein domains between catalytic sites. Secondly, there is no current method that can predict these regions. Now, this is the flow of our predictive model. So, given a protein sequence, the amino acid sequence, we generate four numeric properties. The four properties include the propensity to form structured domains, the propensity to form non-disordered regions, and we also use two indexes measuring the propensity to form certain secondary structures. By using the four numeric properties, we generate four features by using a window centered on each residue to be predicted. The four features are fed into a logistic regression model and to output the score for each residue to be a DFL. The unique part of our predictive model is that the four numeric properties are empirically selected to be relative to our prediction. Another unique part is that we use a window to generate the features. For each of the four properties, we generate one feature that combines all residues in this window, in contrast to other approaches which use individual residues in each window. Because now there is no current method can predict DFL regions, so we compare our method with other alternative methods. Include the UMA method, which is used to find flexible linkers, FL, and 5 methods to find flexible residues, F, 3 methods to find disordered residues, D, and we also include one domain predictor. We assume the regions which are not predicted as domains on linkers, L. We also combine the predictions of flexible residues and the predictions of disordered residues. In this case, we find the residues which are both disordered and flexible, DF. We measure our prediction quantity by the area under ROC curve, the AUC value, and the ratio. The ratio quantifies how much better the predictor is than a random predictor at the low range of false positive rates. We focus on the low range of false positive rates because we have more non-DFLs than DFLs. So by considering the low range of false positive rates, we are not over-predicting the DFLs. So from this table, we can see that our method has the highest AUC value of about 0.27, and the ratio of about 3.3. So this means our method is about 3.3 times better than random in the low range of false positive rate. Our method is also significantly better than other methods. We use the p-value 0.01 to as a significant level. Our method is also characterized by a very fast runtime. In this plot, we compare our runtime with other methods, including the UMA method, to find the flexible linkers, and the predeflexy to find flexible residues, and one disorder predictor, the ISCB. In the x-axis, we have the average length of proteins. We divided the proteins in our training and test set in different groups by their lengths, and we record their average runtime per protein for proteins in these groups measured by milliseconds in logarithmic scale. So this red line is a runtime for DFLpred, which is our predictor. So we can see our prediction has a low runtime when the proteins are longer, and when the proteins are shorter, our runtime is comparable with the ISCB, which is one of the fastest predictor of disordered proteins. So this method, the DFLpred, can be considered as a high throughput method, which means it is able to perform genome-wide prediction in under a day. The fact is that this predictor can complete the prediction of the complete revealed human proteome in just under 1 hour. So this is one big advantage of our method. Now, since this method outputs the probability score for each residue to be a DFL, to interpret this score, we plot this figure. So in the x-axis, we have the probability score, and in the y-axis, we have the fraction of native DFLs which are predicted to be below the given score, and the fraction of native non-DFLs in the blue line. The native DFL is the red line. So in the blue line is the native non-DFLs which are predicted to be below a given score. So by using scores 0.07 and below, we can find half non-DFLs from here, half non-DFLs and 20% DFLs. And by using scores 0.12 and above, we can find half DFLs and 20% non-DFLs, because in this case, about 80% non-DFLs are below this score. So in other words, by using scores below 0.07 are more likely to be non-DFLs, and scores above 0.12 are more likely to be DFLs. Our method is provided as a web server in this address. Users can copy and paste their query sequence in this text box. They can also upload their sequence file by this button. The DFLpred outputs probability score for each residue to be a DFL. We use a cutoff value 0.18 to assign class label. So if a residue, the score is above 0.18, we assign this residue as a DFL. Otherwise, we assign this residue as a non-DFL. DFLs are indicated by the uppercase letter, and non-DFLs are indicated by lowercase letter. We accept up to 5,000 protein sequences at one time, so users can perform multiple predictions at one time. Now I will demonstrate our prediction by using an example. This example concerns the troponin I protein, and I'm looking at the N-terminal of this protein. So this part of the protein, in the bottom, I have the residue number. This green line is a native annotation for protein domains, and this the purple line here is the native annotation for disordered regions, and the red line is the native annotation for the DFL regions, which we want to predict. So from here we can see that the second domain, the troponin I domain, has two subdomains, the IT arm and the regulatory head, which are connected by this DFL region. The two subdomains are connected by this DFL region. And for the first subdomain, the IT arm, it has two hexes, and the two hexes are connected by this DFL region. So this DFL region connects the two hexes. This curve here is a score from DFLpred. So we can see our method has high probability score for these two DFL regions. So we can find the two DFL regions accurately. Now, if we take a look at the score from the UMA method, which is a predictor to find flexible linkers, we can see this method has high score for the N-termini of this protein. It can somehow find this region in between the two domains, but it cannot find the two DFL regions. Now, if we take a look at the score from the prediflexy, which is a predictor for flexible residues, we can see the score from prediflexy fluctuates widely among the protein sequence. This is because flexibility is a property for individual residues instead of for the longer regions such as DFL. So this method cannot be used to find the DFL regions. Now we are looking at the scores from the disorder predictor, the ISPRED. This predictor has high score for the first DFL region and can partially find the second DFL region, but it also has very high score at the N-termini of this protein, which is a disordered region, but it's not a DFL region. So in this case, it produces a false positive. Now we are looking at the score for the threadome, which is a predictor to find domains. We inverted our score. So regions which are not predicted as domains, we assume them as linkers. So we can see this predictor can find this region. It has high probability score for this region, which is in between the two domains, but it cannot find the two DFL regions. Now let me summarize my presentation. So we have the DFLpred, which is an accurate and quick prediction of disordered flexible linker regions in proteins. We have a web server in this address. So finally, I'd like to thank the NSERC of Canada and the Kimenda for the grant to my supervisor and the China Scholarship Council to support my research. And finally, I'd like to thank the ISMB Travel Fellowship and the Faculty of Graduate Studies and Research of University of Alberta to support my trip to this conference. So that's all my presentation. Thank you very much.